Hello students, we are discussing about transcription process. In previous part we have seen that how the transcription process can take place, why the transcription process is known as heterocatalytic. In this part we will discuss on how the transcription process may occur in prokaryotic as well as eukaryotic cell. First of all, during transcription, one enzyme that is called RNA polymerase. RNA polymerase is the enzyme which help in transcription process. This RNA polymerase enzyme is made up of 5 units, and those 5 units are 2 alpha units, 1 beta dash unit, 1 beta unit, and 1 omega unit. RNA polymerase enzyme is made up of 5 units. These 5 units are 2 alpha unit, 1 beta dash unit, 1 beta unit, and 1 omega unit. If the RNA polymerase has only these 5 units, this type of RNA polymerase enzyme is called core enzyme. What is core enzyme? The RNA polymerase enzyme which contain only these 5 units. So that type of RNA polymerase is called core enzyme. 2 more units are also attached with the RNA polymerase, then the name of the RNA polymerase is changed. Suppose when RNA polymerase enzyme, it means it has already 5 units attached with sigma unit. The RNA polymerase enzyme which is attached with the sigma unit, this type of enzyme is called hollow enzyme. What is the difference between hollow enzyme and core enzyme? Core enzyme, only the RNA polymerase enzyme which has only 5 units. If the RNA polymerase enzyme attaches one another unit, that is called sigma unit, this is called hollow enzyme. Is that clear? Hollow enzyme. Why the sigma unit here? What is the role of sigma unit? This sigma unit, first of all, it initiates the transcription process. As the DNA converts into RNA, the process will start due to the sigma factor in sequence manner. If there is no sigma factor, then this core enzyme may be transcript the DNA piece, but not in sequence. How the transcription process occurred in prokaryotic organism? So we can say that transcription in prokaryote or prokaryotic cell. We have seen that in transcription, DNA change into one strand of DNA change into RNA. Suppose we have 2 strands of DNA. If this is contain 3' to 5', this strand contain quadratic, and another one is 5' to 3'. And this process may be started. Before starting this process, we need what is core enzyme, what is the difference between core enzyme and the hollow enzyme. Firstly, we will take the hollow enzyme. Why? Because the RNA polymerase only recognize the site of promoter from where the process starts. This is the role of RNA polymerase. And the sigma unit, it initiates the process of transcription in a sequence manner. So we have already stated there are 3 more units are also help in transcription: promoter, structure of gene, and terminator. Suppose this is the promoter end. A promoter is attached with the 5' of upstream coding strand. This is the coding strand, this one is template strand. 5' upstream, and another one is at the last, it is called terminator. This one is terminator, and this one is promoter. Clear? First of all, RNA polymerase. What is the role of RNA polymerase? RNA polymerase only recognize the site from where the transcription process starts. The RNA polymerase attaches at this site with promoter. This is the RNA polymerase. If we attach RNA polymerase coenzyme, this enzyme will transcript the DNA one strand. It means the another strand will be occurred from transcript RNA change from this one side, but not in a sequence. So, on node coenzyme we attach a promoter at the hollow enzyme because RNA polymerase with sigma factor. This is called hollow enzyme. The first process is recognition and initiation. Because RNA polymerase recognizes the site from where the transcription process starts, and sigma factor is responsible for initiate the transcription process in a sequence manner. First of all, the hollow enzyme attached with the promoter and the transcription process starts. In a sequence manner. As the process initiates, the sigma factor follows. As the process initiates, it means transcription process starts, sigma factor follows. One another unit attached with the RNA polymerase, and that is called rho factor. It means second stage. That is called elongation. As the transcription process starts, sigma factor follows. You can say that transcription process starts and sigma factor follows. Second, elongation. After falling the sigma factor, one another unit that is called rho factor. It means here it contains the rho factor. What is the role of rho factor? Rho factor elongates the process of transcription and finally termination. It means there are two functions of rho factor: elongation the process and termination the process. Elongation, as RNA polymerase contain rho factor, the transcription process elongates. It means formation of RNA is started. It elongates. And finally, if this RNA polymerase reaches to the termination end, as the RNA polymerase reaches to the terminator end, rho factor follows or detach. Rho factor detached, then the RNA polymerase is also follows. So that at terminator end the process will termination, means end up, stop. Third stage is termination. The elongation process continue till then the RNA polymerase contain rho factor. But as the RNA polymerase reaches to the end of the DNA strand, or you can say that terminator end, the rho factor detached from the RNA polymerase and RNA polymerase also follow. So the process of transcription stop. That process is known as termination. At the terminator end, the rho factor detach from RNA polymerase and And RNA polymerase is also fall off. So the process stop. So the process of transcription stop. And this is called termination. It means one strand of RNA which is made up of this one like that is attached to the RNA polymerase. This RNA piece is formed out. This is the transcription process occurred in prokaryotic. As we have already known that the prokaryotic cell, that cell which doesn't contain nucleus membrane. So, in prokaryotic cell, transcription as well as the next RNA to protein translation process, both the process occurred in cytoplasm. It is different from the eukaryotic. Here this the RNA it is also called nascent RNA or hnRNA. That is from hnRNA. Again listen, in prokaryotic cell, either the process of transcription or translation, both occur in cytoplasm. There is no nucleus membrane. But in eukaryotic, how the transcription process occurred in eukaryotic? We know that in eukaryotic cell there is nucleus membrane. Transcription process occurred inside the nucleus, but translation occurred in the cytoplasm. So the hnRNA which is synthesizing nucleus comes out from the nucleus membrane to cytoplasm. So there is need to capping and tailing, splicing some unwanted, remove the unwanted proteins. How this process occurred? Transcription in eukaryote or eukaryotic cell. As in previous part we discussed in prokaryotic cell, one type of RNA polymerase enzyme synthesize two or more proteins. So it is polycystonic. But in eukaryotic cell, RNA polymerase enzyme of three types: RNA polymerase 1, RNA polymerase 2, RNA polymerase 3. The RNA polymerase 1, which only synthesizes one type of protein, that is rRNA. It means RNA polymerase synthesizes DNA one strand into rRNA. RNA polymerase 2, it synthesizes or it transcribes the DNA one strand into hnRNA, heterogeneous nuclear RNA. What is the full form of hnRNA? Heterogeneous nuclear RNA, or mRNA, messenger RNA. And RNA polymerase 3, this type of polymerase enzyme synthesizes one strand of DNA into the tRNA. It has many units such like as snRNA. snRNA is called smaller nuclear RNA. scRNA, many more. So, in this we have seen that there are different type of RNA polymerase enzyme. And one type of RNA polymerase enzyme synthesizes only one type of protein. So, in transcription in eukaryotic cell is a monocystonic. This is the first difference. In prokaryote polycystonic, in eukaryote monocystonic. Another difference is in prokaryotic, whether the process is transcription or translation, both occur in cytoplasm. Whereas in eukaryotic, transcription occurred in the nucleus and translation occurred into the cytoplasm. So the hnRNA which is synthesized in the nucleus comes out through the nucleus membrane into cytoplasm. But there is no any irregulation or irregularities occurred. Some more process after these process, three more processes also occurred. The first process after the formation of hnRNA, first process that is called capping. What is capping? As we have seen that, suppose this is the double-stranded DNA. It contains 3' to 5', 5' to 3'. RNA polymerase enzyme which synthesizes the hnRNA. This one line. This is the nascent RNA. And always translation, transcription, replication, this process always occurred from 5' to 5' to 3'. So this is the 5' of hnRNA. There is no irregularities occurred in hnRNA. First process is capping. After these three processes, because in these three processes hnRNA is synthesized. After this one in eukaryotic, one more process, three more processes. First of all, capping. Capping means to cap the 5' end with a chemical substance called methyl guanosine triphosphate. It means in this process, methyl guanosine triphosphate chemical substance is attached as a cap towards the 5' end of hnRNA. Methyl guanosine triphosphate means GM triphosphate is attached to at the 5' end of hnRNA with the help of methyl guanine enzyme. Methyl guanine. This is the first process as a cap, so it is called capping. Why capping is produced? Because as the hnRNA comes into the cytoplasm from nucleus membrane, there is no irregularities can be found. This is the first process, capping. Second process is tailing. As the hnRNA descend, means nascent hnRNA descends from the RNA, it contains this one line. This is the 5', this one is the 3'. 5' attached to the capping. The third prime end is also attached with some residues of adenylate with the help of poly A polymerase. So there is no changing can be occurred in this towards the 3' end. Some residues of adenylate or adenylate, means 200 to 300 are added with the help of an enzyme called poly A polymerase. This is called tailing. So there is no change occurred towards the 5' end and 3' end. And after that, some unwanted protein which are synthesized during in the hnRNA. There are two types of protein. One is express coding or coding sequence, express sequence that can be seen in the mature stage are called exons. And one type of protein is unwanted, which cannot be, which is known as unsequenced, or you can say that unwanted sequence, unexpressed sequence called introns. Introns are not used, unwanted end. So we remove the introns and add the exons. That is called splicing. Third phase is splicing. In this process removal of protein, you can say that unexpressed sequence or unwanted proteins are introns. Remove the introns and addition of exons. Exons are wanted sequence, express sequence which can be used in mature stage, and these are not used in mature stage. These are non-functional, these are functional. So non-functional units are removed and functional units are attached with the ligase enzyme and removed with the help of some special protein. Addition of exons with the help of ligase means addition of exon with the help of ligase and removal of protein. To remove protein, which one protein? Introns. To remove introns, we use snRNA with protein complex. Protein complex that is called smaller nuclear or smaller nucleus ribonucleoprotein. Introns are removed with the help of snRNA with some protein complex which is commonly called smaller nucleus ribonucleoprotein. This is also called SNRNTS, or we can say that SNRs. This the whole process occurred during the transcription process in eukaryote. Before eukaryotic transcription, these three processes, after these three, capping, tailing and splicing is also take place. But in eukaryote, prokaryote, the transcription process occurred by the three phases. This is the major reason between the prokaryotic transcription in prokaryotic and eukaryotic. In next chapter, next part, we will discuss on translation process.